lesson. You're watching the lesson on the microscope. So we're going to be talking about, well, the microscope, the parts involved, and what they do. So, what are microscopes for? Microscopes are used to find very small and sometimes seemingly invisible things. So here we have a microscope and a picture of what the microscope might be looking at. And if you paid attention during the previous lessons, you may recognize these to be a type of cell. Well, what kind of cell would they be? Well, they're very kind of blocky, aren't they? And they have that nucleus there. Well, it must be a plant cell, right? Because you see the nucleus, and it looks very blocky. It's definitely a plant cell. And so, microscopes are used to look at small and seemingly invisible things. So, how was it made? A man named Anton von Leeuwenhoek invented the first microscope to look at very small living things. And so it looked like this. And it really was only the size of a sunglass lens. And so he would place it over his eye and he would look at things. So this is the hole he would look at. So if I were to draw a very bad eye, it would look through this small pinhole, and whatever he was looking at was right there. And so he'd place it really close to his eye, and he would look at very small things. And so this was arguably one of, if not the first, microscope made by Anton von Leeuwenhoek. And so this is what he looked like. And using scrapings from his teeth, so basically he took a toothpick, scratched his teeth a bit, and put the stuff he scraped off onto his microscope. And he saw living things. He saw things moving around. He gave them a name. He called them animalcules. Right? It almost sounds like molecules and animal put together. And that's what he noticed them to be. Very small animals, so he called them animalcules. At the same time of Anton van Leeuwenhoek looking at his teeth scrapings, a scientist by the name of Robert Hooke made his own microscope and looked at cork. And when he looked at cork, he saw these. He saw these sort of blocky shaped things. He saw these little boxes. He thought they looked like rooms. So he called them cellulae, meaning little rooms. This is why we have the name cells, because cells, to Robert Hooke, looked like little rooms, and so he named them that. Over time, scientists became more and more aware of cells and found out that, well, cells are everywhere. And if something is alive, well, it must be made out of cells. And so the theory that came from this is called cell theory. Cell theory. And so cell theory basically explains where all life can be found and what constitutes what constitutes life, basically. So we have bacteria, protists, algae, us, right? Everything that is living is made out of cells. So cell theory has three big rules. Everything that is living is made up of cells. Cells are the basic or smallest unit of life. And all cells come from other cells. So the first rule is saying all living things are made up of cells, right? If you're alive, you're at least made up of one cell. And we've talked about this. Two is that cells are the basic or smallest unit of life, in the sense that once you get smaller than a cell, it's no longer alive, right? So the smallest something can be, and still be considered alive, is a cell. And all cells came from other cells. So this video will talk about that. And this will be a sort of small activity we'll be doing in class, filling in the review of Anton van Leeuwenhoek as well as Robert Hooke. So, how do microscopes work? Well, nowadays there are two main types of microscopes. Something called a compound light microscope and an electron microscope. So you can see how big the electron microscope is, and you can probably make the assumption that we won't be using an electron microscope. We'll be focusing on the compound light microscope, but we will briefly talk about the electron as well. So, compound light microscopes. These are the common ones you're used to. They work in a very similar way the human eye works. It works very similarly to the human eye, right? So the human eye, one of the most amazing organs we have. It takes light and allows our brain to make images, right? What's the smallest our human eyes can see? So I'll be asking that, and it's roughly around 0.1 millimeter. So the human hair is about 0.7 millimeters wide. And so that's what the human hair would look like under a microscope. So if something starts to get smaller than this, it starts to get a bit harder to see. It starts to get a bit harder to see, unless it's brought very, very close to our eye. And so the microscope is modeled after how the human eye works. So how does it work? The eye allows us to see things by focusing light using our lens and cornea and having it hit the retina. So we have the cornea, we have the lens, and in the back, we have a retina. Microscopes work in a very similar way. So, the parts of the microscope. You need to know the parts of the microscope. It'll come up again and again and again in science, and so knowing it now is good for knowing it later. The eyepiece. So the eyepiece, this right here, you can see it right there, the eyepiece. This is the part you look through. This is where you put your eye, right? And it has a lens, typically it has a lens that magnifies things by 10 times, right? Another name for eyepiece, or at least the lens here, is called the ocular lens, right? So the eyepiece is what you look through. It has a lens that magnifies objects by 10 times. And another name for it is the ocular lens. This is where you put your eye. This is what you look through. Then you have the tube. So this is the eyepiece right here, right? Now we have the tube. The tube allows the light that is being magnified from the objective lenses travel to the eyepiece. So it allows light to get to the eyepiece. We'll talk about the objective lenses in a minute, but basically the tube allows light to get to the eyepiece, or to get to the ocular lens. It's a tunnel that allows light to travel to the end of the microscope. The coarse focus. So the big knob, not the little knob here, this big knob moves the stage up and down to focus the image. It's usually used at low magnification. So it's called the coarse focus. And what you do is when you move that big knob, it focuses things in very big changes. It creates these large changes in focus that allow you to see things at low magnifications, right? So the coarse focus changes the focus of the image as you turn the knob. The fine focus, which is this little boy right here. Oh, it's a bad drawing. Just fix that. This little knob right there. The fine focus moves the stage slowly to focus the image. So the coarse focus causes these big changes, and the fine focus makes these small changes. And so you use them together so that you can get a nice focus, right? If it's really blurry, you would use the coarse focus to get a big change, so you can get closer to a proper focus. And if it's just a little bit blurry, you'd use the fine focus to make just small changes so that you can see the image or whatever you're looking at just a little bit better. You have the arm, it's right here, right? So you have the ocular piece or the eyepiece. You have the tube. You have the coarse focus. You have the fine focus attached to it. And here is the arm. This connects the tube, this is the tube, to the base, and this is the base. We'll talk about that as well. It's also where you hold and carry the microscope, right? You put your hand here, and you put your hand on the base, and that's how you carry it. And that's it. So the arm is connecting the tube and the base. The revolving nose piece. It's this disc up here. And this disc's job is to spin the objective lenses. So again, this is the second time we've mentioned objective lenses. And so just a quick preview. These are the objective lenses. So when you're changing from lens to lens, you spin the revolving nose piece. These lenses, so the objective lenses, right, are attached to the revolving nose piece or the spinning nose piece, and they are used to magnify the image. They're used to magnify the picture. Now the lenses typically go from 10 times, 40 times, and 100 times. So these lenses, the lowest one typically magnifies the object by 10, the second lowest by 40, and the third by 100, and sometimes you'll have an extra one by maybe even 500 to 1,000. The stage, well, like in a play, the stage is where you put what you're looking at. So the object being viewed is placed on the stage, right? There's a hole in the center of the stage where light hits the object, but the stage is where you put the object to be viewed. The condenser. This is the part under the stage, and it directs light to the stage. So light will be coming into the microscope, and the condenser focuses the light under the stage so that it goes through the hole. The diaphragm. The diaphragm is below the condenser. So you have the stage, you have the condenser, and you have the diaphragm, right? And so the diaphragm controls the amount of light coming through. The diaphragm controls the amount of light coming through. So when you turn the diaphragm, it's usually like a disc. It'll make the hole in the stage bigger or smaller. It sort of works like our iris does, right? If you stand in a very bright room, your eyes, your pupils might get smaller because your irises are making your pupils smaller so that you don't collect that much light, so that you don't go blind, you know? If you don't have that ability and you walk into a bright room, everything might seem too bright. You might not be able to see anything. Well, the same is true with a microscope. If you open the diaphragm too wide and there's too much light coming through, the object might just disappear in the light. So you may need to make the hole smaller on the stage. So you turn the diaphragm. And the opposite is true. If you're in a dark room, well, your iris in your eye will make your pupils larger, right? So you can collect more light, so you can see more things. Well, the same with the diaphragm. If you turn the diaphragm so the hole is larger, more light gets through, and so you can see more in a dark image. And then we have the light source. Naturally, this is where the light comes from. And so you need to know all these parts of the microscope as this loads. You need to know the eyepiece, the tube, the rotating nose piece, the arm, the objective lenses, the stage, the coarse focus, the diaphragm, the fine focus. Now, sometimes the fine focus will be on another knob, or it'll come out. There'll be another knob coming out of the coarse focus, and the source. I may even ask you what the base is. That's the base, right? And that's it. That's all you need to know for the microscope. So now we're going to talk about how the microscope is able to magnify everything we're looking at. Well, as light passes through the stage, the light is magnified by the objective lens. So we have the light source. It hits our sample on the stage. And so the objective lens focuses the light onto one point, magnifying it. And so what happens is we have another lens that flips the image, because when you magnify something, typically it'll flip it, so it'll look upside down. And so another lens will just flip it again so that we can see it. So as light passes through the stage and the object, the light is magnified by the objective lens and the eyepiece. So both the eyepiece and the objective lens magnify the image. So if you recall, the eyepiece magnifies the image by 10. So automatically, the image is going to be magnified by 10. Well, depending on the eyepiece, the objective lens you use, you will get an extra bit of magnification. So the lowest objective lens is 10 times, and the eyepiece is also 10 times. What is the magnification? Well, you looked at an object, the objective lens magnified it by 10. right? So it's 10 times larger. Well, then the eyepiece magnifies it by 10 again, which makes it 100 times larger, right? So 10 times 10. So when you look at something at the lowest magnification, usually you're looking at it at 100 times because the objective lens magnifies it by 10, and so does the eyepiece. The middle objective lens may be 40, right? 40 times. So you're already looking at it at 40 times, but then the eyepiece does another 10 times, which makes it 400. So you're looking at something 400 times the size. So when you look through a microscope, you'll see something like this. And the area, or what you see, is called the field of view. And this can be centimeters to even a millimeter to even less than that. And so the field of view will always look the same through the microscope, but because of how much you're focusing on it, the distance might be from 1 centimeter all the way down to millimeters or even smaller. So I said I'd briefly talk about the electron microscope. The electron microscope uses electrons to generate images. And so it needs all this machinery to basically shoot electrons at things, and as the microscope picks those up, it creates an image. And so it doesn't necessarily just use light, but it uses electrons. And we won't be using this, we won't really be talking about it too much, but it is a way to use a microscope and see things much smaller than what a compound light microscope could see. So it lets us see even smaller things, up to tens of thousands to even a million times magnification. So hopefully that was useful, and I'll see you next lesson.